protein structure, an application of like, well, let me back up. We talked about how you have, in proteins, you have the polymer, which is the polypeptide, and the polypeptide is made of the monomer amino acids. You have the 20 different amino acids. Remember, you don't have to memorize the structure, just know which ones are hydrophobic and hydrophilic. So let's say you have a primary structure, which is like the beads on the string part, right? Where you just have all the amino acids linked together. Peptide bonds hold the amino acids together. Remember that? Okay. Now, the secondary structure, that's where you have the alpha helix, the beta sheets. Tertiary structures, when the secondary structures fold up into a more like three-dimensional model. And then you get the quaternary structure where you get multiple tertiary structures. Remember, you can just have only like a tertiary structure for a protein, meaning there's just one subunit that makes up that whole protein. But most proteins, like say hemoglobin, you have 4 subunits that make it up. Okay? Now, hemoglobin is a protein that carries oxygen. Well, in your red blood cells make up your blood, right? One of the big parts of your blood. They carry the oxygen to all the cells throughout your body. Inside the red blood cells are all these proteins called hemoglobin. Hemoglobin is really good at binding oxygen. Okay? Now, there can be a mutation that happens where you swap at the number 6 amino acid. You should have glutamate or glutamate, and instead you swap that for valine, the amino acid valine. And you're like, okay, doesn't seem like that big of a deal, but that one change swaps the entire, makes the entire subunit not be shaped properly. It will now have the sickle-like shape in the subunit, where when you get a bunch of those sickle-shaped hemoglobins all together, they will then form, ultimately, this sickle-shaped red blood cell. And the consequence of this is this red blood cell, there's 2 consequences. One is it doesn't carry oxygen as well. And the second is, having a nice sickle shape like that, why would that be an issue for like a blood cell? Blood cells need to carry blood throughout your whole body. They move through arteries and veins. They're like kind of tubes. Think of drains, and if you have long hair, be my hint here.
Blood clots.
Yeah. Blood clots, yeah. Having this sickle shape, not only does it not carry oxygen as well, it clots more easily. Whereas having that nice ovalish kind of shape, that's not going to clot as easily, and it carries more oxygen. So this is not good. You don't want this. So there you go. Basically an example of like how things can go wrong. And again, the point is one change can make big differences. And that change is in the primary structure, just changing the amino acid sequence. Okay. Okay. Now, proteins, like you have your normal protein where it's folded up nicely. Proteins can fall apart, basically unfold. We call that denaturing. Okay. There's a bunch of different things that can cause denaturing, like proteins are finicky. They want a certain condition to like operate properly. So like that's why your body temperature needs to be at like 98.6. If your body temperature gets too high, the proteins will denature. They will fall apart. And then remember, a key function of proteins is as enzymes. So those chemical reactions that we have to be doing in our cells wouldn't be able to happen properly. Now they can renature, like if you get back to the proper conditions inside the cell. So temperature is one of those conditions. pH, so the hydrogen ion content. So your blood wants, your cells want to be right around that pH of 7. Temperature, pH. I'm forgetting one of the other ones. Those are of salt concentration, right? Don't want to get too salty, you know. But yeah, those are gonna be your 3 main ones you want to know that cause protein denaturation. So there you go. And yeah, to be clear, denatured protein is biologically inactive, meaning it doesn't work properly. Okay. Questions on any of that? Cool. All right, last little bit here is protein structure. So if you're kind of curious, like, hey, how do they figure out the structures of these proteins? Basically, they will turn them into crystals. So you can get crystals of proteins by basically getting all of the water outside the cell, and then you'll isolate the proteins from the other parts of the cell. Then you'll then take all the water that's surrounding the protein or is inside the protein, and then you're left with these crystals. And if you shine a beam of X-rays, this isn't an X-ray, but like kind of like if you were to shine like the light, like my laser pointer at the crystal, that light will then diffract. And if you have a screen there, you will then get a diffraction pattern. And if you do some, you're trained in this, right? Certain proteins, like based on how they diffract, they move, they spread apart the X-ray light, that will tell you the actual structure of the protein. So if you change the shape of the protein a slightly different way, you're going to make the diffraction pattern look slightly differently. So there you go. That's the last slide here. Call it X-ray crystallography. And it's worth pointing out that some proteins, as they're going through that primary, secondary, they're beginning to actually fold up into their final three-dimensional shape. They will go through different shapes before they're at that one stable shape. Would be what is the functional shape it has in the body, like the shape that it's going to have when it's doing its job as an enzyme or a structural protein or whatever different jobs proteins can take on in the cell. Yep.
How fast does it do that?
Oh, get to the final stable shape? No clue. I'd have to look it up. Probably seconds, maybe. Pretty quick, right? On a chemical level. But I'd have to take a look at it. So, anyways, all right. Other questions on proteins? Okay.